Well, what is next generation sequencing? So I'm pretty sure everyone is familiar with what DNA and genome is, right? So sequencing in general, it's the process of figuring out the order of DNA in the genome, right? So uh, sequencing, it started when Frederick Sanger, he started working on genome sequencing. Now, how does that help us? So once this started, things started getting out of control, and then sequencing has improved by leaps and bounds over the last few decades. And nowadays, whole genome sequencing can be done almost within a day. Now there are different types of there are different types of sequencing. The first generation, as we call it, that's the one that was started by Sanger himself. So they initially used chemical degradation methods, and then they would just track the small, like the nucleotides through gel electrophoresis. So that was the first generation sequencing. Now, following that, we had second generation sequencing, which has been there for almost 2 decades now. And in this, the technology had developed even better. So sequencing of DNA fragments could be done parallelly, and there was higher yield, so better quality of sequence. The cost was relatively cheaper, but we still could do only short reads. So what are short reads? We'll take a look at that later. But for the time being, that's second generation sequencing. And recently, uh, they have also developed third generation sequencing. So here you can sequence very long strands of DNA. In fact, you can sequence single DNA molecules without having to stop in between the reads. So the second and the third generation sequencing are what we call next generation sequencing. So it's also known as high throughput sequencing. Now, these are some of the types of NGS, next generation sequencing technologies that are being used. So for short reads, that's the second generation reads, we have something called ligation and another technique where there is synthesis. Now, what are short reads? Short reads means basically when you sequence the DNA, you have to, chop it up into small pieces initially. So short reads means sequences of DNA that could be up to 150 or 300 base pairs. So 150 to 300 nucleotides long. So those are short reads. And long reads are very long stretches of nucleotides or DNA or RNA, or as we call them, reads. So they could be up to even 10K base pairs. So that's a lot of information right there. Now, the two types that we see here, ligation and synthesis. So what happens in ligation is that the principle of NGS by ligation, so it involves detection and incorporation of bases, all right, nucleotide bases by DNA ligase. So what ligase will do is it'll take nucleotides and then stick them to, you know, and elongate them so that you can find out what the underlying sequence is. And this happens in a very specific manner. The detection is generally done using some form of fluorescent label. The one, let me just turn on my laser pointer. Yeah. So this is a label through which people can detect which nucleotide is being attached, and so they will find out exactly what is the sequence here. And an example of ligation, one of the NGS equipments or the technique that uses ligation is solid. So solid is sequencing by oligonucleotide ligation and detection. So basically that's what it is. Now synthesis, NGS by synthesis, it uses 4 fluorescent labeled dyes. Again, we are using labels here, but here what we do is we use 4 of them. So for each of the best pairs that we know, A, T, C, and G, and then we track the addition of these labeled nucleotides as the DNA chain is complementary copied. So each time this goes and binds, it'll come sit, emit one of the lights so that we can know what the underlying sequence is. Now, when we talk about long reads, third gen, this is relatively new. So what we can do is we can perform real-time long reads. So these are new techniques where a single molecule of DNA or RNA can be sequenced in real time, all right, without the use of all these fluorescent labels, or we don't even have to do amplification in this case. So an example is the Oxford nanopore. Um, it's an example of this technology. So what happens is you have long fragments of nucleotides, right? DNA. And then these are passed through a protein nanopore. And then the changes in electrical current, they are monitored as the nucleotide passes through it. So you can see here, this is being recorded, and that's how you're able to find out what the nucleotide is or the sequence is by decoding the electrical signal. So that's a very brief introduction on the current NGS technologies that are being used. So, and what exactly are the applications of NGS? Well, there are several applications, in fact. The first one is genome assembly. So genome assembly, where we will perform de novo whole genome sequencing analysis. De novo whole genome means the genome of whatever organism that you want. It could be something that you have discovered. It could be some unknown species. So in that case, what we do is we reconstruct the genome from scratch. That's what de novo means. So genome assembly, it aims to reconstruct genome of some species that are, unknown in nature. And many genome assembly projects have been completed till date. Of course, we already know of the Human Genome Project for one. And then we have reference genome of several species. And since Human Genome Project is one of the milestones of NGS, what it has contributed is that with the Human Genome Project, now researchers can find out exactly how many genes are there in the human body and what are the type of genes that are present, where they are present, etc. The second application that we see here is genome variations finding. So this can be done through whole genome sequencing. What happens here is that the human genome is 3.2 billion base pairs. So that's a lot of information. Now, everyone on this earth, they share 99.9% of their DNA. Everyone is quite similar. But what about the 0.1%? So that there is a chance that 3.2 million nucleotide bases, ATGCs, can be different between different people. So that's, again, a whole lot that can be different among human beings. So since the genome of each individual is different from that of the reference genome, so reference genome in this case, say the one that we have sequenced from de novo whole genome sequencing, so it won't exactly match with that of the reference. And these genomic variations, they can cause many diseases. There are a whole lot of diseases that can be caused due to variations. And these variations, we call them SNPs, SNVs, and so on. I think you'll be learning about this in tomorrow's lecture. So I won't go into much detail. Now, because of NGS, through sequencing, we can now identify what are the variants in each and every single individual. So this could be used for so many purposes, like in the field of medicine. This can you can go for personalized gene therapy and personalized medicines. These are all concepts that are not widely used, but these are all the applications of NGS. Now, uh, sorry. Yep, sorry about that. So now what about the other applications? So reconstructing the transcriptome. transcriptome is basically the entire total sum of your mRNA. mRNA, as we know it, are messenger RNAs, which are then converted into proteins, right? And proteins are essential for the functioning of a normal human being or any organism, as a matter of fact. So every human cell has the same genome, well, more or less, right? So now these human cells, we have different tissues, and they express different set of genes at different times. Now this set of genes expressed in a particular cell type is called the transcriptome, like I just said. And then we can use NGS to measure the expression of genes. How does that happen? That is what we will be looking at in today's workshop. Now, finally, another application that goes hand in hand with RNA sequencing is decoding the transcriptional regulation. So this is done using chip sequencing. That's another type of sequencing technique. Now here we have proteins, right? There are certain proteins called transcription factors. What they do is they regulate the expression of genes. So that means how a gene is turned on or turned off. That is exactly what gene expression means. And transcription factors are those proteins that control how the gene is being turned on or turned off. So sometimes when a transcription factor fails to bind to the genome, the corresponding gene target, it'll also fail to express. Or in some cases, it may, just start expressing without turning off at all. And so the cell cannot function properly. Now, it's important for us to understand how exactly this works, and this can be done through NGS. So these are some of the applications of NGS in brief. And before we dive into RNA-seq, these are some terminologies that you'll definitely come across when you perform NGS analysis. So let's just get into it. Now, there are things called replicates. Okay, so what are replicates exactly? When you perform an experiment, you always have to compare it with something else, right? If you want to perform an experiment, you certainly have to do some statistical analysis. And you cannot perform a statistical analysis using only one sample. You need more than that if you want to calculate all the mean and variation and stuff like that. So whenever you perform any NGS analysis, you always have experimental replicates, copies, multiple copies of the same thing. Now, there are two types of replicates. What is the technical replicate and a biological replicate? So technical replicates, it basically means, yeah. So say you have a mouse. You have only one single mouse and you take different samples, or you take out the DNA sample from this mouse and you put it into the 3 vials. So that's a technical replicate. That means you're repeatedly measuring the biological sample from one single sample. And then biological replicates, we have 3 distinct samples here, and we will measure the amount of DNA, or we'll just try to figure out the sequence, or which gene are being expressed by taking the DNA from different mice. So one for each mice, 1 vial. So there we have biological replicate. So biological replicates is the one that we generally use, right? So why is that? Now these are collected from biologically distinct samples, like I said, and so statistical tests can be applied to evaluate the differences. Now, when we average across distinct replicates, it helps in precise measurement of gene expression, for example. So generally, biological replicates are the type of replicates that are preferred when you are trying to do any sort of experiment in NGS. Now, we've seen how replicates can affect the study and the outcome. Now let's take a look at how groups are divided. So this one is quite straightforward. We have something called control and treatment. So the control group is something that does not receive the treatment, or is a standard for the treatment. So it's like, when you perform a lab experiment or something in wet lab, you take water as the control, right? And then you will compare that with some other chemical. So that's the control group. Now, treatment group is the one that receives the treatment. And this is the group that researchers are actually interested in. So this could be anything. This could be a sequence where a particular gene has been knocked down, or this could be the treatment group could be a cancer cell and the control group could be a normal cell. So that means you're going to be comparing the cancer with the normal cell. So we have two distinct groups, control and treatment. It could be anything. You can rename it as, again, knockdown versus whatever. But the fact remains that control and treatment is the general term that is used for two groups that are being compared with each other. Now, there are some more terminologies. I explained about reads earlier, but I didn't exactly say what reads are. So what a read is it's just a fragment of DNA or RNA that is sequenced by the instrument. So we have a long sequence here. Imagine this is your genome. We don't actually sequence it in this way, right? So initially we'll cut it into small pieces. That's what short reads are. So this is a read. That means we'll have, say, around 150 or 300 nucleotide sequences, and that'll be a short read. So reads are just a fragment of DNA that you want to sequence. And the read length is something it specifies the length of the read, so 150 base pairs, like I said. And the red things that you can see here at the end, these are called adapters. Adapters are just short sequences of DNA that are ligated to the end of the fragments here. This is done prior to sequencing. And it's performed during the library preparation step. So I won't go into much detail about all these things, but just know that when you sequence it, when you sequence the DNA, you need something for the DNA to hold on into, so that it doesn't just fly away during the sequencing. So this is the reason why we attach adapters to it. And yeah, and then we have something called depth coverage. So depth coverage is the measure of number of times that a specific genomic science site is sequenced during a sequencing run. That means when you sequence something, you don't just, read it once and quit, right? What happens is if you want accurate readings, you try to read it multiple times. That's what depth of coverage is, or we just call it read depth also. So here is an example. You have a reference genome aligned to it. These are your reads. This is a read depth of 4. So you just specify it as something, a numerical value, and then followed by an X. This one has a read depth of 1, of 3, and I think you understand what I mean, right? So now let's take a look at the different types of reads. We have single end reads and paired end reads. Single end means you just cut the DNA and the machine will read it from the 5' to the 3' end. That's it. You just do it once. Now in paired end read, what happens is once you cut the sequence, it will do the sequencing from 5' to 3' from one end, and then in the other end it'll again perform the same thing from 5' to 3'. So this is done so that you can get more accurate readings. So paired end reads are generally more expensive, but this is generally more reliable, so as to say. Now there is an inner distance between read 1 and read 2 that we can see here. That means it's just a gap that occurs whenever you perform paired end sequencing, and it generally occurs in all types of paired end sequencing. So these are some of the terminologies. Now let's go ahead with RNA-Seq. So what is RNA-Seq then? RNA-Seq or RNA sequencing is the measurement of RNA in a given biological sample. Okay, so we take out the RNA and then we sequence it, and then we try to find out how much RNA is present at specific regions of the genome. So this way what happens is you can measure if a certain gene is, expressing more, or is it being repressed. So in a manner of speaking, that means either find out whether the gene is turned on or off. So why do we need RNA-seq then? It's used to identify genomic loci, um, the expressed cell in the, in a cell or a population at any given time over the entire expression range. I think we'll see all of that in detail. Maybe things might be a little confusing right now. And it's a quantification of expression differences between distinct conditions. So what am I talking about? Consider a normal cell. Okay. So we take a normal cell from a normal human being, and then we take a cancer cell from a cancer patient. Now imagine that both these individuals or both these cells, they have a gene called gene A. Now what happens is, um in the normal human being, in a normal cell, gene A could be functioning, pretty well. There's no problem. It's being turned on and off whenever it is required. In the case of cancer cell, what happens is that gene A no longer turns on, or even if it does, it's being repressed, or it's been turned on for a much longer time than what is necessary. So this is the reason why we do RNA-seq, so that we can find out the difference between the normal cell and a cancer cell with respect to the expression of genes. And that is over the entire expression range. That means not only for gene A, we will try to find out how much gene is being expressed uh, in, for all the genes that are present in that cell. Now, how is RNA sequencing done? Initially, we will isolate cells or tissues, and then we will extract the RNA. Uh, we can extract rRNA, ribosomal RNA, or messenger RNAs. Okay, and generally mRNA is what we use when we talk about RNA-seq. Now we will isolate and fragment specific DNA. So suppose you want mRNA, you will isolate it from the rest of the debris and the other DNA and RNA. You will isolate the specific one of your interest, and then you will just cut it into small pieces. Once that's done, you will convert it into cDNA, due to various reasons, for storage stability, because RNA is not exactly a stable molecule. So we will convert that to cDNA first. Then we will construct a sequencing library, adapters I was talking about. So we're going to add adapters to that, and we're going to get it ready for the process of sequencing now. And then we will do PCR amplification, polymerase chain reaction, so that we can create multiple copies of this of the library, and then we will perform the actual sequencing. Right. So now that is understood, um, this is what we're interested in. So how do we analyze RNA-seq? Now you have a bunch of data that you want to analyze. How do you exactly do it? Um, the steps that are provided here, these are very general steps. They're always used in RNA-seq analysis. Of course, you can add several more stuff to this, but we'll stick to this one right now because it's quite easy, to be honest, to understand and to follow. So initially, the data that you have, the RNA, there's lots of cleaning to be done. Like it's just a raw product that's been exported from the factory. So you will perform a quality control to that. Quality control could be trimming of adapters. So those adapters that we have seen earlier, sometimes, they don't get removed from the sequence. They get stuck somehow. So we need to remove those adapters, filter out some of the qualities. So we'll take a look at this further on. And then we can also filter the length. We will generate a quality check report, or a quality control report. Now, once you take care of the quality, we will align it. So remember, all the DNA or all the RNA, they are in pieces. So what you will do is you will use one of those reference genome like the ones that has been sequenced in the Human Genome Project, for example. And then you will lay them next to the reference and try to find out exactly what the original sequence was when you took it out of the organism. So alignment is done using the target genome. That's the that's the term for your sequence. That's the target. And then a reference genome, a genome that is already been sequenced, and we already know the order of that sequence. And then you will get an alignment report. Now you will go for read counting. What is read counting? So we know that some genes are expressed in cancer type, whereas it's not being expressed in normal type, or vice versa. It's being expressed in normal, it's not expressed in cancer type. You will find out what the reads are for that. Um, we'll take a look at this in more detail later on, but for now, we will count the number of genes or the number of reads that are aligned to genes. That's done using feature counts, and then we will also annotate them. So right now we don't know which read belongs to which gene. So we're going to find that out through annotation. Uh, that will give us a report. And then finally, we will perform what is called differential expression analysis. Differential expression analysis means you're going to find out which gene between the control and the treatment are differentially expressed. So is it being overexpressed? Is it being underexpressed? That's what we're going to find out. So these are the major steps that are generally used when you perform any type of RNA-seq analysis. Now we're going to take a look at all the steps in close detail. Yeah, so FASTA, FASTQ. I think most people are familiar with the FASTA format, right? So a FASTA format, it always has the header, a description of the sequence, followed by the sequence, right? Now what the FASTQ has, it's very similar to the FASTA, but just that in this case, we have a queue here. What does this queue mean? It's what happens here is that in the FASTA, you will store the quality of the sequence that is being generated. So it generally starts with an @ sign, with the information of the read that you have sequenced, maybe the equipment name, the length of the sequence. Then obviously it's followed by the sequence that you have generated. And then there could be an additional plus followed by the same thing all over again, just so we know that it belongs to the same read. And then finally, we have a weird combination of characters here. So these are all ASCII characters. And what each one of them represents is the quality of the nucleotide that we see here. So G corresponds to 3. Um, there's a lookup table, which I unfortunately do not have at the moment. What it will show you is exactly what 3 means. So 3, it's just a placeholder, um, the for the algorithm or the tool to know what exactly is the value stored. So it could go from 0 to 1,000 or from 0 to 100. Um, all of these they store values, how well the sequencing has been performed. So are we sure that this is a G? If we're say 100% sure, perhaps we can say we can assign it a score of 3. What about the next one? If we are say 50% sure, it'll be assigned a plus. So that's how it works. So moving on to quality control. What is quality control? Now, modern high throughput sequencers, so high throughput means next generation sequencers, they can generate tens of millions of sequences in a single run. Okay. Now, before we analyze the sequence, we always perform quality checks, like I mentioned before, to ensure that the raw data looks good. There's no problem. There's no biasness in your data, because this can eventually affect the outcome of your analysis. Now, uh, yeah, what does this show you? This will show you the range of quality values across all bases at all positions. Okay, fine. So we have positions in read. So here we can see that these are the number of reads. So one strand of read can have up to, in this case, clearly up to around 100. So This is the length of your read. This is what we call a Fred score. So this is the score according to which we measure how accurate our reading is. Now, how this is calculated, we'll take a look at it in the next slide. But for now, what you have to understand is the colors here. We have green, we have something that looks like orange, and then we have pink. So what we want is our data to always fall under this category. So this is the part where we say, okay, the data is fine, it's of good quality. Now the yellow or the orange region is the part where things get a little, we can say that it's okay, but we could have done much better, but then it's acceptable. If it falls, if the score is, you know, at least 20, we can say that it's acceptable. We can move on and perform an analysis. But if it falls under this pink region, poor quality, so then we'll have to perform all the steps that I mentioned here, like adapter trimming, filtering, lens rate filtering, and so on. So the Fred score. So what exactly is the Fred score? For looking at this, let's take a look at the table first. So if we say uh, if Fred assigns a quality score of 30 to a base, that means if a nucleotide ATGC is assigned a score of 30, that means that there's a chance this base is called incorrectly is 1 in 1000. So what does that mean? Let's tally it with the table. So for a Fred quality score of 30, uh, that means we know with 99.9% accuracy that the nucleotide we have assigned, it could be A, T, G, or C, whatever it may be, it's 99.9% accurate. That means we're 99.9% sure that the nucleotide is either an A or T or G or C. Now, what about 20 then? Because remember we said 20 is in that acceptable zone, but it's not actually good. So that means base accuracy is 99. We're 99% sure that this is the sequence that we have generated. So anything that is less than 20, so look at 10 here, it's only 90. Doesn't seem like a lot, but that's a very wide margin of error, which we generally do not want. So anything that falls below a Fred score of 20 is something that we will discard or try to filter out. Now, a quality score can be used to describe the chances that a call was correct or incorrect, right? And again, we have seen that they will assign a confidence of this much. So these are all are pretty good, right? So the probability is 1 in a million for a given alignment. Now, the Fred score, what is it exactly? It's just a score that is assigned to the nucleotide to estimate the probability of error. So that means that the base is called incorrectly. So what we can calculate the error probability using this equation here. So Q means your Fred score or Q score, Fred score, let's call it. The formula is simply minus 10 log to the base 10 of probability of error. So if you try to do the calculation, you'll end up with this. And fortunately, we don't have to do any calculation because the quality score is automatically generated when we perform the sequencing. That's how we know what the scores are. We don't actually have to calculate it manually. So when we do sequencing, the output that we get, the raw output, it's usually in FASTQ format. So we have already seen what FASTQ format is. Now, we've already seen this plot here. Let's discuss what exactly are the contents of this plot. So I don't know if it's visible or not, but there are some blue lines here. There are red lines, and then we have these things that look like T's and inverted T's. So this plot, the yellow boxes, these are called box whisker plots. And the element of these plots are the red line that you see here. If you follow the laser dot, you can see some red lines. These are the median values. So median, as in statistics median. And then you have the yellow box, which fill up a majority of the plot, uh, this represents the interquartile range. So interquartile range means 25 to 75% of your data, it lies within the yellow region. And then we have the lines here. These are the upper and the lower whiskers. So the remaining, percent, so 10% and 90% of the other data, they lie within this region. So we just have to follow the yellow line, the yellow boxes, and also, of course, the blue line, which represents the mean. So the mean of the quality is this. So just by following the blue line and perhaps even the yellow one, you can tell how good a sequence is based on its quality. So here again, we see that everything is well above 30, which is of good quality. Now, I've already mentioned here, but uh, the x-axis, you can see that it's not uniform. 1, 2, 3, 4, it goes up to 9, and then suddenly they'll just clump everything together, right? So it depends on the length of the read. So sometimes it can be 50 over here. Sometimes it can be 150 over here. It looks like it's around 100. So the positions, the number of base positions that are quite similar, they may be binned together. So like, for example, 18 to 19. These are all the regions that are between 18 to 19. And, yeah. So moving on. Now, what the quality of the sequence is expected to look like. I've already seen this example. The quality is pretty good. Now, what about this one? So this is okay. It's not okay. Why? Because a majority of it falls in the red region, and that's pretty bad. Now, how exactly do we overcome this? Actually, the tools for all the trimming processes will be discussed later on in the second half during the hands-on session. But for now, yeah. So now remember the mean here. If you take a look at this line or any line, as a matter of fact, be it the good quality or bad quality sequence, you see that there's a trend here. It just goes up and then slowly it goes down, right? Or it could just, it just keeps going down. Why does this happen? What causes this drop in quality? So the drop in quality is because of several reasons, which are beyond our control, unfortunately. This is often anticipated. So it's nothing to be worried about because this generally happens when you perform sequencing and it's due to technical reasons. So two common reasons are signal decay and phasing. So signal decay, what happens is, uh, remember the dyes we use, like the labels, the fluorescent labels. So as you go on extending the reads, the fluorophores may degrade, so it no longer emits light. So that means you no longer know what the actual sequence is, and because of that, some ambiguity may arise, and the quality may drop. The second thing is clustering. The clusters are no longer elongated. So when you sequence something, you do it in clusters, right? So what happens is sometimes the primers or the nucleotides, they do not come and bind to a specific part of the cluster, and so there will be no light emitted here. There will be no strands attached here, and because of that, you will see some drop in quality or the overall quality. Now, the second type of reason for the drop in quality is phasing. So nucleotides may be added incorrectly, okay, and they may not be removed after signal detection. So this comes and binds here, and after that it doesn't move. So the next time something comes and binds, this all one will be stuck here, and what will happen is A will keep on stacking up, and you'll see errors in the reads. So if the ones below are something else, or suppose this is supposed to be a C, right? So instead of C, you will also find some A's in between, and they will cause some bias. These are all wrong readings, and so because of that, the quality will again drop. This could happen because of incomplete removal of the 3' terminators, and the fluorophores. or incorporation of nucleotides without effective 3' terminators. That could also happen. Now, sequence quality is one of the plots that will be generated later on during the hands-on session. But for now, let's understand what this means. So this is the mean sequence quality, the Phred score. So anything that is above 30, 30 or more than 30 is considered good. We already know that. Now, the sequence, whenever you do a quality check or a quality control, it should lie within this region. So you'll get a very similar plot to this. And what you expect to see is a curve that is almost the same as the one that you see here. So everything should be within 30 to anything above 30. Okay. What about per base GC content? Now, per base GC content, what it shows you is the percentage of bases, the 4 nucleotides at each position across the reads. So again, remember, these are the reads, and these represent the quality of the reads. Now, for whole genome sequencing, the ratio of A and T and G and C should remain the same or relatively same. Now, you see here that for A and T, it's close to 30, okay, the percentage of bases. And then for G and C, it's around 20. Now, what happens is in RNA-seq, within the range of 1 to 15, 10 to 15 here, there is a non-uniform distribution. Well, why does that happen? You don't have to be concerned about that because this thing is quite normal when it comes to RNA-seq, because when we do the sequencing part where we prepare the libraries and then the adapters, there is a priming step during the library production, and we don't actually really know why this happens, but during the roughly 10 to 15 first bases, random priming happens. So because of that, you can see these kind of ups and downs. But this is, again, like I said, this is completely normal when it comes to RNA-seq. You shouldn't see it in any other type of sequencing, but for RNA-seq, this is acceptable. Similarly, the per sequence GC content. So the GC content should be around this region, like say from 40 to 60, some from 30 to 60. In RNA-seq, again, you may get some error here saying that it fails. The quality check has failed because it's greater or less than the theoretical distribution. But again, that is acceptable only in the case of RNA-seq. So these are just some of the quality checks that are unique for RNA-seq. We'll see what the other types are in the hands-on session later on. Now, once you're satisfied with the quality of your data, what you will do is you will perform alignment now. So you have the sequence of your interest, and you have a reference genome, a template, and you will try to put them together. So alignment, sequence alignment, it's basically arrangement of DNA sequences that are similar between sequences in terms of their structure and function. So um, to put it in plain words, you have a box of puzzle. Everything is randomly, mixed up. You have to put them in the correct order. That's what sequence alignment is. And what about the reference sequence? So the definition of a reference sequences is a comprehensive, integrated, non-redundant, well-annotated set of sequences, including genomic DNA, transcripts, and proteins. So imagine these are your RNA reads. Okay, as we can see, these are all cut into pieces and they're all randomly put together. Now, when you have a reference sequence and when you perform alignment, everything will fall into its correct order, and that is exactly what we are doing with alignment. And for alignment purpose, the algorithm that we use has to be very fast. It has to handle SNPs, the variations in the genomes. the insertions and deletions and any sort of sequencing errors that may occur. So some things may get through the quality check, and so we want to make sure that the algorithm is able to handle any sort of errors that we may have missed earlier. So Burrow-Wheeler's transform is actually a technique or an algorithm that is used for several alignment algorithms, not just one, but it's a very popular one, that's for sure. Some of the popular ones that uses it are Bowtie, HiSet, Bowtie 2, the newer version, and HiSet 2 as well. So in brief, read alignment or mapping, as we call it, is the process of alignment reads on a reference genome or a reference sequence, and a mapper or an alignment algorithm. It takes as input a reference genome and a set of reads. And the aim of this mapper or the alignment algorithm is to align each read to a set of reads or the reference genome. So how does this actually work? A very basic illustration of how Burrows-Wheeler transform works. So it's a data compression technique. What it does is it rearranges word characters. Okay, it makes it more compressible. So Burrows-Wheeler transform doesn't actually compress the string or your DNA sequence, but it just makes it more compressible. And this will work in linear time, irrespective of the length of your genome. It's only based on the length of the string that you want to search. So based on the length of the read, the it'll perform the alignment, and this way it's quite fast as well. So we have a short read. This is a read from the sample that we sequenced, and we have our reference genome. So chromosome 1, chromosome 2, 3, and 4. What we do is we will concatenate them into a single string, and then we will perform the Burrow-Wheeler's transform and indexing. So indexing is something like a sort of appendix at the end of a book where you can find certain words and then just go back and see which page it points to. That's, I mean, in a very basic definition of what indexing does. So you look up the suffix of reads. Suffixes, yeah, I don't think I'll get into that for the time being. So you take the short read, and then what happens is from the end of the read, just keep on reading that. You try to look up where exactly G occurs in this Burrow-Wheeler transformed set of reference. If you locate G, then what happens is you will find the next G here, and then the location will keep on changing because there could be several G's, not just one. So it'll just keep on looking. Once it finds that, it'll point to another new direction where AGG happens. And this just keeps on going until you find the complete sequence, or you are able to align the sequence of your interest to the reference genome. So hits identify position in genome where reads are found. So it'll identify the location, and then it'll convert back each hit to the genome position, and then it'll report it back to the user. So this is a very simplified model of what actually happens. So once we perform this, let's see what the alignment actually looks like. So we have the reference here, the very long stretch of strings, and the reads now. Previously, it was all jumbled up. Now it's properly aligned to the reference, right? And just for the sake of context, this is the read depth of the aligned sequence, the one that we talked about earlier. So once we generate the sequence, or once we perform the sequence alignment, it'll generate an output. So initially, when we checked for quality, the output that was generated was still a FASTQ file. We only had a FASTQ file, but once we perform alignment, they're much more, the information is much more comprehensive. So we have all those insertion deletions and various other things that could be present in after the alignment, right? So we save this in a format called SAM, or alternatively BAM as well. So SAM, it's a sequence alignment map format, and BAM stands for binary alignment map. So sequence alignment, SAM as we call it, these are tab delimited text files. They contain the information for each and every read from, after the alignment to the genome. And if you compress SAM into a binary format, that's how you get BAM. And we generally use BAM because you know the size is small, and it allows for indexing, and it's much more efficient when we're accessing data. So without going too deep into the format, let's take a look at what a SAM file looks like. It always has a header file. So a header file, and this is the part where you have the alignment information. So this could mean several things. So the ID of the read, okay, and then we have the flag, which means it'll give you the info on the read alignment. So single, is it a single end sequence? Is it a paired end sequence? So all these columns are actually in decimal number, and 99 actually doesn't look like a decimal number, but you just have to keep on adding stuff like 64 plus something plus something. That's how you end up with 99, which means it's a paired end sequence, and then several other reasons. And then you have rname. Rname over here, it's just reference. This could be a chromosome number. So ID, and then the flag, and then say chromosome 1, chromosome 2, chromosome 3, etc. And then you have position. It indicates the leftmost mapping position of the first matching base within the read. And then you have the mapping quality. It's the quality of the mapping, again similar to your Fred quality score. And then you have the CIGAR score. So CIGAR stands for Concise Idiosyncratic Gapped Alignment Report, which means all the things that we see here are idiosyncratic. These are unique to the specific set of rules, or the alphabets that we see here. So for example, at aim, which means there's a at 8 match, 2 insertions, 4 match followed by 1 deletion, and then lastly 3 match for this sequence, which is nothing but this is the read that is aligned to your reference genome. And then we have a bunch of other stuff as well. Let's not go into detail, but just for your information, this is what the SAM output will look like. So that's great. We have aligned the sequence. We have checked for the quality. Now let's take a look at how reads are counted. We have already discussed about the quality control. We have also talked about the alignment. Now we will be talking about read counting and what it means. So counting reads. Now, once we have aligned all the reads to the genome, the next step is to count how many reads have actually mapped to each gene, right? Now, commonly, some of the tools that we use for counting reads are feature counts, HTC count. Now, all these tools, what they do is they only report the row count. Okay, they report only the row count of the read that maps to a gene at a single location. Row count means we have already seen when all those reads are stacked on top of each other. That's what the row counts are. So for that to actually work, we'll have to normalize that, which we will be doing in the next step. But for now, the input for counting, it consists of multiple BAM files. So remember, once we do the sequence alignment, we generate BAM files. So we'll have not just one, but for every sample. So you have different replicates of the control. Say you have 3 replicates of the control, and for the treatment as well, you have 3 replicates. So once you sequence all those, and then once you do the quality check and generate the align files, you'll have 6 BAM files. So you'll have multiple BAM files, and you'll have something called a GTF file, which we'll see in the next slide. So that's the 2 input that is required for counting reads. Now, the genomic coordinates of where the read is mapped, which means from the BAM file, right now, we just know that it's aligned to a reference genome, but we don't actually know where the genes are in that region. So what it will do is the genomic coordinates of the map region of the reads are cross-referenced with the genomic coordinates of features that you are interested. So features could be things like your introns, exons, genes, transcripts, all those things. So these are all present in a GTF file. So it's something like your reference file, but this one is specifically made for features. A GTF file format looks like. Again, without going too much into the detail, you have the chromosome, and then the source from where it was taken, and then you have the feature. So it could be an exon, it could be a gene, it could be an intron, a transcript, whatever it is. And then the start and end location of each of these features. So in chromosome number 1, it starts from this region and it ends at this region. So what do we have here? We have an exon, and this exon number is this number 1, and then the name of the gene is DDX11L1. And then similarly, you have other stuff here. So these are all exons, and you can see that this is exon 1, 2, 3, but these are all exons of the same gene, and that is why um, they all have the same ID here, the same gene name, and even they even have the same gene ID and the transcript ID as well, just that the exon number keeps on changing. So depending on what it is that you want, if you want to see the entire exon, then you can go for exon features. If you want to see genes, you can select genes as your feature. Again, more detail in the hands-on session. So this is what the general feature format, sorry, looks like. A GFF, or we can also call it a GTF file. Now, how does the counting of features actually work? So when the tool, like feature count, whatever it is, how does it assign what feature should go to which read of yours? So suppose you have ref, so that's actually your read, and sorry, I mean you have something of gene A, and then you have this ref here. What it will do is it'll check how closely it is aligned, and then it'll assign it a feature of gene A. So to your read or your already aligned sequence, you will say that this belongs under gene A category. Similarly for this one also, and so on. So even here, there are two genes here, A and B. However, it is being assigned to A only because there's no overlap of your read with any of the regions in gene B. So these are clear-cut assigning of features based solely on the raw read counts. Now, some things can become ambiguous, which means we don't know exactly where to put them, and so this may be discarded by tweaking some of the parameters in the tool. So we have here that the scenario we were talking about earlier, we have two genes, and somehow our read falls under both regions, the region of both these genes. So this is ambiguous, same thing, ambiguous, and ambiguous. So this is how it's assigned, and this is what it'll look like. So we can see the row count here for gene A, B, and C, some hypothetical genes that we have drawn. And the row count that I was talking about, it's just the number of reads that are assigned to each and every one of these. So this exon has a row read count of 8. This one has 2. Say this exon has 0. This one has something else. This one has 4. Likewise, that's how it goes. So wherever there's no feature, you'll assign it a 0 because there's no read available there. If there are 4 reads, you'll assign a 4, a 3, and generally we don't do it in 2s and 3s and then go up to thousands. But there's a very good chance you'll see some 10s, some 5s, etc. Depending on how strongly a gene is either expressed, there is no expression, and all that. So once you perform the counting of features and the reading of, the read counting process, you'll get an output that looks like this. So the output of the tool feature counts look like. KD stands for knockdown. That's what I'm assuming. You have two samples. Presumably there's a KD1 as well, which is somehow lost in this figure. So we have three replicates of KD, knockdown gene, and then we have three replicates of overexpressed. Now, if you look at this gene here, we see that 57 is the row read count in KD2 for this gene, like the one I showed you in the previous figure. In some, you'll see only zeros and ones. In some, you'll see it in terms of thousands. So that's how the row reads look like. And just by looking also, somehow you can see that there's a difference between the reads of certain groups and between different replicates. So within group and between the groups as well. But this is not a good way of measuring the difference because there are biases which we have to get rid of first. So we'll take a look at what exactly those biases and problems are. But for now, this is the kind of row count that is needed to find the difference between the control group and the treatment group. So knockdown in this case and overexpression. So we're just comparing two groups, and row read counts are the precursors for finding out differentially expressed genes. So with that knowledge, we'll move on to the last step, which is differential expression analysis. Here you will find the genes that are differentially expressed in whatever condition it is that you're looking for. So in the treatment, if you find which genes are expressed, you'll be comparing treatment versus control, or if it's something else like knockdown or overexpressed, like we've seen in the previous table, that's how you'll be doing it. Now, without further ado, what is differential expression analysis? So the textbook definition of differential expression analysis is you take normalized read count and you perform some statistical analysis to discover quantitative changes in expression levels between experimental groups. Okay, so in simple words, that means you want to find out if a gene is expressed or not. Now, before you can perform the actual analysis, the row reads that were generated by feature counts, they have to be normalized first. That is the essential step I was talking about. And normalization is the process where you scale those row counts to allow better comparison between samples. Now, there are different types of normalization techniques that can be used. So CPM, counts per million, is one that is often used. RPKM, FPKM, so read or fragments per kilobase per million. So all these are used. However, some of these methods are no longer being used due to several reasons. There are some certain biasness attached to it. TMM, however, it's used even now in some of the tools. I believe in EDGE-R, it's currently being used for finding differentially expressed genes. So it means trimmed mean of M values. So this accounts for sequencing depth, transcript length, so all these things. Now, why is normalization important? This is a pretty good example. We have two conditions here, condition 1 and 2. We have three genes, so three sets of genes for both the conditions. But one thing is slightly different here is that condition 1 only has 12 reads, whereas condition 2 has 36. So 4 here, a lot. So there's 12 reads here, only 3 here, whereas there's 9 here, and then 5, whereas there are 15. So just by looking at it, you might think that condition 2 is more expressed as compared to condition 1. However, that is not true because condition 2 simply has 3 times the reads as condition 1. And that could be due to several reasons. Um, Sometimes what happens is due to sequencing read depth. We've seen that already, right? So the expression is actually same inside the body, but when you sequence it, when you sequence the DNA or RNA of your interest, some things may be sequenced a lot more. The read depth may be more. So that's how you end up with this. And row read count is something that'll just show you these numbers like 4, 3, 5, 12, 9. So to bring them all to the same level, that is the reason why we have to perform normalization in the first place. So normalization has two problems associated with it. One is to adjust for different library sizes. One is to adjust for differences in library composition. So libraries, those are things that we have talked about early on when we talked about how RNA sequencing is done. But let's take a look here. Just like the previous example, this has sample A has only this much reads, sample B has double the number of reads, say. So why does this happen? I've already explained that this is due to sequencing depth, right? Now, what about the other type of problem? That's library composition. Differences in library composition happens when the number of reads are the same here, but where it is assigned to, it's different. So A1BG, let's compare it with this example. So it's 30 here. Now, instead of 2186, it's 635 here. And these two reads are the same, just that they're assigned to different places. Now, what happens is A and B, they have the same number of reads, even when A2M in sample B is 0. So there is no way of comparing that, right? So for that, that's the reason why we need normalization. Now, how we perform normalization. So the method I'm going to explain now, it's based on the tool DE-Seq. So DE-Seq has its own internal parameter for normalization, unlike the CPM, RPKM, FPKM, and DMM that I explained before. And it's quite simple, to be honest. What we do is we take a small sample here for the purpose of our workshop, and then we have three distinct samples. So we have a list of genes, 3 genes, and we have 3 samples here, and these are the raw read counts for each gene. What we do is we're going to calculate the log values for each of these. So log of 0, infinity, log of 10, log of 4. You keep on doing that, and then you calculate the average of each row. So you just calculate the average here. You'll get infinity for infinity. For this row, you'll get 1.7. For this row, you get 4.3. Now, compare these two genes, A2M and A2M, after taking the average value of logs and before taking the average value of logs. Now, the average here is 96. Whereas, once you calculate the log values, you see that it's actually 73.7, right? This is an outlier because this value is excessive. But once you calculate it to log, it doesn't seem as much as before. So average that is calculated using log, it's smaller, and it's also not as easily swayed by outliers. So because of this single 200, the average has changed by a lot. But once you calculate it, once you calculate the log value and then you do the average, things are what it should actually be, or actually how it's supposed to be. So this is the step: calculate log values, then calculate the average of each of these row. Then you just remove the genes that have that infinity attached to it. Now what you do is you subtract the average log value from the log count. So the average log value that we calculated earlier, you'll just subtract it to each one of your row and column values. So you'll get this now. So after subtracting, this is the output that you'll be getting. Using that, you will calculate the median of the ratio of each sample. Okay, you'll be calculating median of this, and once you calculate that, you will convert the median to normal numbers to get the final scaling factor for each sample. Scaling factors, we'll talk about that in detail. So this is the scaling factor, exponent of minus 0.9, 0.4. So once you get the scaling factor, you will divide the original read counts, the one without the log transformation, by these scaling factors. So 0 divided by this, and then 10 divided by 0.7, and then 4 divided by 2.5. You will do that for each and every row and column. And this is the new scale read count. So if you compare these two, original with the new one, you see that some things are slightly different. The most jarring one perhaps is A2M, where initially we see that 33, that's quite low value, 55 is okay-ish, 200 is a lot. But now after scaling it down or normalizing, now sample A instead of 33, it's actually supposed to be 83 in comparison. And then 55 has gone up to 79. And the outlier that was creating a lot of problems is actually almost equivalent to 80 only. So that's how you do it. So instead of just comparing the row count values, you scale it down first, bring everyone to the same level, and then you carry on with the analysis. So what next? You have already done that. So we take the normalized read count. We're going to perform some sort of statistical analysis to discover the changes in expression between experimental groups. We've already seen that. Now we're going to decide uh, whether a gene that we find uh, and it has and we observe differences in that gene, we're going to see if that is significant or not. So how do you determine what gene is significant, or how do you determine if a gene is differentially expressed or not? So we're going to use a value. It's called the log fold change. So fold change is the change in expression level. So is something you're comparing two things. So one is in 0, the other is in 10. So we can say that the second one is 10 times more than uh, the one that is 0, or something like that. So that's basically what fold change will tell you. And this is in log to the base 2 format. And then we have another parameter that is p-value, with which several things are attached, like fold change is related to p-value, and more or less FDR itself is the p-value, but it's an adjusted version of FDR of p-value, which will which is um, which is found through multiple tests. So this is good for finding out false positive values that may occur by chance in your differentially expressed data. So this may be a little bit confusing, but I promise you I'll deal with all these terminologies later on. So just to understand, we look at the fold change if you want to see if a gene is expressed or not, and we look at the p-value. Okay, so what does all this upregulation and downregulation mean? Let's example, we have 10 sets of genes, same to the previous one. Now this is a normal cell, and this is a cancer cell. So we have gene A, B, B, and C. Everything looks similar, just that uh, we have to tally it with this graph here. So this stands for gene A, B, and C. It's quite self-explanatory. There's no change of expression in gene A. Why? Because let's assume that this is normalized for the time being. We see that the reads are equal throughout gene A in both the conditions. That means the difference between them is absolutely zero. So there's no change in expression. That means the gene um, in normal and cancer, or if you're comparing cancer with normal, there is no significance as far as this gene goes. Now, for gene B, things become a little different because the read or the normalized reads are higher here as compared to your normal cell. And because of that, the value of this goes up. It goes way up. And that's what we mean by overexpression or upregulation. That means the gene is being expressed a little bit more than what it should actually be in a normal cell. So that is overexpression or upregulation. That means the gene is being regulated upwards. Now the opposite thing can be seen here, gene C, and instead of it going up, in cancer, gene C is being reduced as compared to gene C in normal cell. So this should be how much C is. This is how much gene C should be expressed in a normal human or a normal cell. But in cancer, we see that it's not being expressed properly. So that's what underexpression or down regulation means. It's being regulated downwards, or the expression is being suppressed. It's underexpression. So overexpression and underexpression are the terms that are used when we talk about differentially expressed analysis. Like, for example, how many overexpressed genes did you find, or how many genes were downregulated? What's the um, you know what's the p-value for or what's the FDR rate for the over expressed genes that you find. So these are some of the things that are generally discussed when you talk about differential expression analysis. And how this is actually done is a little more complicated than what I just explained. Um, One of the popular tools that is currently being used is DE-seq. So DE-seq, what it'll do is it'll model the row counts using normalization factors, size factors, to account for differences in library depth. So remember that normalization step with the tables, which I just explained some few slides back, that's actually what DE-seq does. That's the method used in DE-seq. So we have already seen how it normalizes the data, um, and then it'll estimate the gene-wise dispersion. It'll shrink these estimates. So what this means, I'll just tell you. Um, estimating size factor. So you model the row count of the gene, which we have already done. Then you're going to do the shrinking part. You're going to test for differential expression analysis. So initially, after you normalize your after you normalize the data that you have, you're going to estimate the gene-wise dispersion. Okay, so normalization step. Let's start at the beginning. What it does is it'll allow accurate comparison of gene expression level between samples, right? So it'll help you identify differentially expressed genes that are truly biologically relevant rather than just reflecting the technical variability. Now, once you have the normalized data, you will perform a step called the gene-wise dispersion estimation. So gene-wise dispersion estimation, it estimates the degree to which the expression level of a particular gene differs among the samples in a data set. So this could be within the data set or between the data set, which means you have, let's take the previous example, 3 control samples and 3 treatment samples, right? So obviously we're looking for differences between control and treatment, but we also have to consider the fact that there could be differences between the 3 samples of control and differences between the 3 samples of the treatment itself. So within the sample and between the samples. And this is why we perform gene-wise dispersion. We're trying to find out degree of expression levels of particular gene among the samples. That's what it is. And by dispersion, it means the spread or variability of the data. And spread or variability doesn't mean your variance, because dispersion is not variance. It's directly related or directly proportional to variance, and it's inversely proportional to mean as far as statistic goes. So once you estimate the gene-wise dispersion, I guess you all know about curve fitting. So what we're going to do is we're going to plot a nice graph where we'll have the mean and the variance to find the dispersion, something like that, and then you're going to fit a curve to that. So you'll see the output of what the curve looks like during the hands-on again. So different genes, they have different scales of biological variability. Now, but over all genes, that means all across the genes, there will be a distribution of reasonable estimates among the dispersion. Now, curve fitting, what it'll do is it'll help us estimate the expected dispersion value for a gene of a given expression strength. And we will then shrink the gene-wise dispersion. This is done by, this is done in order to reduce false positive rates. So false positives, for those who don't know, false positive means something that is actually wrong, but sometimes it'll be considered as true because of either the p-value, and there are different reasons for that. So something that is actually not, but by mistake we consider this the correct one. So we're going to shrink the gene-wise dispersion, which we estimated earlier, in order to reduce false positive values. And genes that are low in dispersion estimates, they're shrunken towards the curve, and then the ones that have higher or that are more accurate, and then the higher shrunken values are outputted for fitting into the model, and differential expression testing, which then brings us to shrinking log changes. So basically what it means is you're just going to find out what the log fold change is between the different conditions. So like the normalized count of your normal divided by normalized count of your treatment, for example. And once you do that, you will finally go for the testing. So what happens here is we use a certain test called the Waltz test. It's a statistical technique where you will set forth a null hypothesis. So a null hypothesis in this context is there is no differential expression between the two groups. So when you compare the treatment group with the control group, the null hypothesis will state that these two groups are the same. There is no difference between them. Now, uh, what we will do is we're going to compute the probability value between the two groups that we call the p-value we talk about. So what will happen is when the p-value is small enough, so there is no cutoff for that, but let's say a p-value of 0.05, which is the standard p-value often used for this kind of analysis. When the p-value becomes small enough, we can reject the null hypothesis. So the null hypothesis says that there is no difference in expression between the treatment and the control. But once we reject that with a low enough p-value, we can say that no, there is actually a difference between the treatment and the control. So that's the importance of p-value. It helps us, to accept or reject the null hypothesis. So anything that accepts the null hypothesis will be discarded because we're not interested in that. We just want those genes that reject the null hypothesis and say that these genes are expressed. Now, when we say p-value of 0.05, that means there's a chance that we'll still get, there's a 5% chance, in fact, that we'll get false positives. And since p-value is calculated for each and every gene that we have, that means, the greater the more the genes we have, the greater the false positive rate. Like, for example, if you have around 3,000 genes, we have way more than that, but just assume that we have 3,000 genes that are expressed. So roughly that means based on a p-value of 0.05, at least 1/3 of our gene is a false positive. So that's a huge amount. Now, how do we overcome that? So we can do something called a multiple testing. So what it does is it'll actually help you reduce the number of false positives. So DE-seq, it has its own inbuilt multiple testing method. It borrows that from the Benjamin-Hochberg method. So it'll calculate the thing called a false discovery rate. So false discovery rate. It's again very similar to the p-value because it is a derivative of your p-value. And in general, we set a cutoff of 0.05 again in this case, which means the number of false positives now. We cannot completely eradicate all the false positives. That's not, I'm not saying it's not possible. I'm just saying that you'll end up with very less number of genes in that case to work with. But what it'll do is it'll remove most of the ones that are false positives. And FDR and p-values can be adjusted according to your needs. It could be 0.05, it could be zero point 0.1, or some people like to use 0.01, 0.005 if they want like really good accurate results with very low false positives. So that's how DE-seq works in a nutshell. Now let's take a look at what the output of DE-seq, or not exactly DE-seq, but of differentially expressed genes look like. So we'll understand that with the help of a thing called a volcano plot. What a volcano plot does is it'll plot the fold change. Okay, the fold change in the middle, there's a 0. It can go up or it can go down. That means a gene is expressed or it's being repressed, or overexpression and underexpression. Then you have the minus 10 of minus log 10 of the p-value. So we're looking at the adjusted p-value or the FDR that we just talked about right now. So anything that is less than 0.05, we're going to consider, and any fold change. If you see a fold change that is greater than 2, I don't think I mentioned this before, but for the fold change also, we have a certain cutoff. So any changes that we see that has a log fold change of greater than 2 is something we consider significant. So remember I told you why log fold change and p-values are important, because these are the only two things that you will generally look at when you look at the output of any differential expression analysis. That is, what is the fold change? What is the p-value? Is it greater than 2 or greater than or less than, depending on whether it's overexpressed or underexpressed? And what is the p-value? So p-value of adjusted p-value of less than 0.05 is something we should aim for. So just as an example, you can see RBP1 is a gene that is expressed a lot. And then we have another gene called CSN1S2B, which is pretty much downregulated by a lot. So these are the top two genes in the context of this volcano plot that we see here, the result of a differential gene expression analysis. So all in all, this is the kind of result you can expect to get once you perform differential gene expression analysis. Now, before we talk about what else we can do with this, let's just quickly recap the steps in case you have forgotten. Data retrieval. Now, data retrieval is something which I haven't actually talked about in this topic, because there are numerous ways through which you can get the data. It's not actually possible to sequence the genome by yourself all the time, or say you don't have enough funds, you just want to work on something else, you don't have the cell lines. There could be numerous reasons. So there are actually different ways of getting the data. Perhaps one of them is directly from NCBI. I'm sure everyone's aware of NCBI. So from NCBI, you can retrieve the data from the SRA archive or from the GEO database. So these are just some of the resources through which you can get data. Now, once you get the data, it's still in its raw format. So you will perform quality check. After checking the quality, you will perform the alignment on the reads. Once you perform alignment, you will quantify the reads, and you will annotate them according to the gene name. And then once this is done with the raw reads, you will perform normalization, and then you will find genes that are differentially expressed. So that is all the steps as far as analysis of NGS data in RNA-seq goes. So what now? We have a large list of genes. Once you do your expression analysis, you will get a few thousand genes at least. Now, what do you do with that information? How do we extract biological meaning from it? Now, one of the commonly used, and I would say this is a very standard step, standard next step in this process, that is to perform functional enrichment analysis. What we do here is we're going to look at where those genes are associated with, like which biological process is it associated with, or inside the cell, where is it found? Is it in the cellular membrane? Is it somewhere else? And also we want to, we're interested in the functioning of it molecularly, as in what does it do? Like, is it a part of some process? Does it do ATPase activity, for example? So these are collectively known as gene ontology, and gene ontology is a structured, controlled vocabulary that we use. It's for classification of gene function at a molecular and cellular level. And then the three subcategories that GO is divided into two is your biological process, the molecular function, and cellular compartment. So you can specify any one of these parameters, and then what you can do is find out where the gene lies, and which function it's a part of. And then, of course, since this is a statistical test, again, the p-value will come into play, but this is generally how it is done. Now, we're towards the end of this presentation for now. For the next session, I will just give a brief introduction on the tool that we will be using. RNA-seq using Galaxy. So Galaxy, some of you might have heard of it, some maybe not. So Galaxy, it's a web-based platform. Sorry, it's for computational biomedical research. So you can perform various activities using Galaxies. Of course, it's it's open source. All the tools that are present in Galaxy are, the standard tools that we generally use. Like it's not like they make their own tools and try to publish it. These are all standard benchmark tools that are available in Galaxy. And the best part is you have an attractive GUI. So I'm pretty sure most people are, not comfortable with coding or, writing commands just to run all the tools, and you might be scared of how to get started with it. So Galaxy is a very good platform for you to start learning NGS as a whole. So there are numerous positive points of Galaxy. I guess you'll see that uh, by yourself when we go on with the hands-on session in the next part. That will be the end of the first session.